Hello, grade 11 students. In this video, we're going to talk about the semester 1 exam review worksheet. Please keep your worksheet in front of you in order to start talking about the topics and the solution for this worksheet. This worksheet is going to cover the biological diversity and uniqueness of individual, chapter 1, pages 10 to 20. Also, it's going to cover chapter 2, DNA, genetic information and the cell cycle, pages 21 to 39, and also chapter 3, protein synthesis and enzymatic activity, pages 41 till 51. Also, you have to review the worksheets and the notes on your copybook. Question number 1. Indicate the true statement or statements and correct the false one or ones. If it's true, simply you say true. If it's false, you have to say false and corrected incomplete answer correction. So replication of DNA occurs during the DS phase of the interphase. So this is true. Helicase is the enzyme that unwinds the two DNA strands. Also, this is true. C. Tetraid line up in the middle of the cell at metaphase 1. This is true also. 4. Silent point mutation, it changes the sequence of the DNA or the sequence of the amino acid. False. Silent point mutation causes no changes in the sequence of the amino acid. 5. mRNA sequence is complementary to the coding, which is the non-template, the non-transcribed. No, it's complementary to the non-coding, so this is false. The enzyme that unwinds the two DNA strands is called the helicase. This is true. Moving now to question number 2. Changes taking place in a cell during its division. The following table shows the variation of the amount of DNA as a function of time in a cell undergoing cell division. So we have time in days 0, 3, 5, 10, 10.5, 11.5, 12, 20, and 30. And we have the quantity of DNA in a bit unit 7, 7, 14, 14, 7, 7, 3.5, 3.5, and 3.5. We want to draw the graph. Drawing the graph, you have to know that the one in the table above will be down and the one down will be in the y-axis. So the time in days will be in the x-axis and the quantity of DNA will be on the y-axis. So this is the graph. So I use here every 1 centimeter in the x-axis that corresponds for 10 minutes, and on the y-axis every 1 centimeter corresponds to 2 units for the quantity of DNA. Remember this, the graph should have a title, have a scale, and should be drawn using a pencil. Analyze the result. What is the analysis? So as the time increases from 0 to 5, the quantity of DNA increases from 7 to 14, and then it remains constant till the 10th day. Then, as time increases from 10 to 12, the amount of DNA decreases from 14 to 3.5, and it remains constant till the end of the experiment. What is the type of the cell division? The cell division, we have 14 AU. It's divided into 2 or divided into 4. It's divided into 3.5. So 3.5, so divided into 4 cells, meaning that this cell division is called meiosis. The number of the daughter cells that are obtained, there are 4 daughter cells. In which type of cells it occurs in the sex cells? Now let's move to question number 3. But question number 3 talking about the retinitis pigmentosa. Retinitis pigmentosa, it's a hereditary disease. It's the main cause of visual impairment, 30% of the visual deficiencies. The disease starts by affecting night vision and reducing the visual field. It's caused by progressive degeneration of the rod cells, which are photoreceptors of the retina containing the protein rhodopsin. To understand the origin of this disease, we study the structure of proteins encoded by different alleles of the rhodopsin gene. The rhodopsin gene, consisting of 1044 pairs, encoded a protein of 348 amino acids. Document 1 showing a portion of the nucleotide sequence coding of the coding strand. So pay attention here. They said the coding strand of the rhodopsin gene, individual with normal and other with retinitis pigmentosa. Pick out from the text the cause of retinitis pigmentosa. They said that it's caused by progressive degeneration of the rod cells, which are photoreceptors of the retina containing the protein rhodopsin. Referring to the genetic code table, so you have to look for the genetic code table which is given. You have to determine the amino acid sequence, and you have to determine the amino acid sequence resulting from the expression of the portion. So the given are the transcribed strand for rhodopsin, so the mRNA is going to be complementary to the transcribed, but the T is going to be replaced by U. Okay? So here, part 3, they start referring to your answer onto part 2 and to the given of document 2 to compare the two nucleotide sequence. So here, the allele of the individual with normal phenotype and that of the affected individual are identical, except at their nucleotides 404, 405. The normal has 2GG, while in the allele for retinitis pigmentosa, they are replaced by TT. So here amino acid sequences, they are identical except at 135th amino acid, where it's arginine in the sequence of the normal and it's leucine in the sequence of the affected. So the modification of the rhodopsin gene is translated in the level of the nucleotide to the level of protein, and this is the origin. So they said here, specify the type of mutation. Here we have substitution of nucleotide 404 and 405, where they are transcribed at the level of mRNA by new codon. These codons will lead to new amino acids. Based on a full comparison and to your information throughout the origin of the retinitis pigmentosa. So these amino acids sequence, this new amino acid sequence, will affect the 3D structure of the protein rhodopsin. This protein will become non-functional. So this protein exists in the root cells, in the photoreceptors. So the change in its function will lead to the impaired night vision, and it will be for the retinitis pigmentosa. Number 4, we have to talk about oxytocin and vasopressin, where they are two hormones. They are synthesized by the post-pituitary gland of the mammals. The first has the uterus as the target organ, and the second has the arteries as the kidneys as the target organ. For this document 1 and 2, they are showing the sequences or the DNA sequence of the basis for oxytocin in 1, and in 2, the DNA sequence for vasopressin, only the non-transcribed. So the mRNA is the same as the non-transcribed, only the T is going to be replaced by U. So we'll have this sequence. So this is the same sequence, by the T is replaced by U. All of them, they have the same sequence, only the T is going to be replaced by U. Compare the two sequences. So we noted that there are 6 differences at the level of DNA sequences. Look here at the position number 7, 21, 22, 23, 24, and 27. So here, as you look, TGC, it's TGC, it's TAC, TAC, ATC here, TTC. So this is the position number 7. CAG, it's CAG, AAC, it's AAC, DGC, it's DGG, CCC, it's CCA. This is position number 21. C here, it's A, T, it's G, G, it's A, and also GGT is GGA at the position number 27. At the level of the polypeptide or the amino acid cis, tire, isoleucine here, phenylalanine, GNN, GNN, ESN, ESN, cis, pro, leo, arginine, and the last one they have the same. So we have only two differences at the level of the polypeptide at position 3 and position 8. 14. Explain why the difference between the nucleotide sequence does not sure affect the sequence of the amino acid, because you have the mutations here, because some types of mutation they are silent, so they don't change in the amino acid sequence, so there is no change in the sequence of these two proteins. That's why we have no change in the amino acid sequence. Please, you have to practice on these and you have to study well for the exam. Thank you.